Good morning and welcome to our webinar today on establishing bioburden alert and action levels. We're glad you're able to participate today. Here to present our webinar are Martel Winters and Wendy Wangsgaard, our resident experts here at Nelson Laboratories. Before we get started, let's take care of a couple of housekeeping items. We welcome all of your questions, and you can submit them at any time. Martel and Wendy will randomly answer as many as they can during our last 15 minutes of the presentation. Martel and Wendy can be contacted afterwards as well with any of your questions. You can reach Martel at 801-290-7864 and Wendy at 801-290-7830. Now I'd like to welcome and tell you a little bit about Martel and Wendy. Martel Winters received a bachelor's degree in microbiology with a minor in chemistry from Brigham Young University. He has been at Nelson Laboratories for 19 years, and he is currently a senior scientist. Mr. Winters serves on many AAMI working groups and is currently the co-chair for the Microbiological Methods Working Group. He is currently heading an AAMI task force for a document regarding environmental monitoring for terminally sterilized medical devices. He is also on the AAMI Sterilization Standards Committee and on the AATB Standards Committee. He is a registered microbiologist with NRCM and a specialist microbiologist. Wendy Wangsgaard has a PhD in pharmacology and toxicology from the University of Utah, where she also received a Bachelor of Science in Biochemistry and a minor in Chemistry. She currently is the Bioburden Department Scientist at Nelson Laboratories. She is also a member of the AAMI Microbiological Methods, Radiation Sterilization, Sterilization Terminology, and Sterility Assurance Levels working groups. Wendy is also a registered microbiologist with NRCM. I'd like to now welcome.
Good morning or good afternoon to those of you who would like to welcome you in joining us today on our webinar regarding establishing bioburden alert and action levels. This webinar is actually based on a paper that Martel and myself and some colleagues here at Nelson Laboratories put together. Information regarding that paper will be provided to you at the end of this. But the people that are not on this webinar that need to be recognized who were instrumental in getting this paper published, Esther Patch. She is now a study director in our chemistry department, and Ashley Ferry, who is now a member of our quality assurance team. Those are our Nelson colleagues. But also, we had a huge boost from Harry Bouchard, who was a statistician at the FDA for many, many years, and he was responsible for crunching the numbers and providing us the statistical guidance. Some of that will be discussed a little bit later on in the webinar, but anyway, thank you to all of them. It was definitely a group effort. So I just want to talk a little bit about why we felt the need to do this paper in this webinar. And as you know, most national and international standards out there talk about bioburden, sterilization, or environmental testing, and they all bring up establishing alert and action levels to assist in demonstrating that you have continued control over a process or a product. So they'll give you guidance, but usually they don't tell you anything more than that. One of the most common practices in the medical device industry does involve using standard deviations to set these levels, and there are some types of guidance out there, such as the radiation sterilization documents that use bioburden to help establish your sterilization dose. And in that case, they'll provide you a table. But if you are using an overkill approach, that's not the same method. And so there are different approaches that you need to keep in mind depending on how your product is sterilized and also, your manufacturing process. So what we wanted to do is just provide you some more guidance regarding that We work in a testing laboratory. We are prone to seeing many, many different types of products and also every type of sterilization modality that might be out there. And so because of this unique perspective that we have, we could use some of this data and our knowledge with working with sponsors and put together this paper and this webinar. So a little background. If you refer to the ISO 11737-1 document, that is on bioburden testing, and specifically in Annex A, clauses A85 through A87. One thing that is constant throughout all of this is that you do need to base any levels that you are going to establish on historical data. You need to gather the data. As I mentioned previously, standard deviations are quite common in using means in order to evaluate your data and then use that information in setting your levels. It's incredibly important to also use your data as trending analysis. And you need to make sure that once you have established your levels that they do remain appropriate in the way that you've established them. And then the last piece of information here is about spikes. Spikes happen. It's a natural part of bioburden testing. And even if your averages stay within the levels you've established, a spike may be an indication of an infrequent problem, or it might be an indication of something that is going wrong in your process. So that's a little bit of background on what you find in the bioburden documentation. Now, what you see here is a standard bell curve. And I am showing you this because whenever people talk about normal distribution, this is typically what they are talking about. You have your average there in the middle, and then most of your data will either be 1, 2, or 3 standard deviations away from your mean. And as it indicates there, you know, that covers 99% of your population. But all of that being said, I will now tell you that bioburden does not fit normal distribution. Part of that we've already addressed when I told you about spikes. If you have spikes in your data, that will definitely skew it, and so it will not fall within that normal distribution. Now, another thing that is often seen in the industry is that your testing will yield 0 CFU. Now, if that's the case and you have no bioburden, you can't even establish a normal distribution or a curve for that matter. That being said, what you really need to keep in mind as you move forward is that you gather data. You know, if you have data that says what your bioburden is, then you are using valid information, very useful information, and that is specific for your manufacturing facilities and your product. And then upmost in that also is the safety that is in your sterilization process. Every modality has a built-in level of safety with overkill methods. You have a 12-log reduction. Even the radiation tables, they take into account the variations that you might see in bioburden, but there is ultimately safety in all of the sterilization modalities. Continuing along that same vein, what we found as we did our review of all of the data that is out there, and we've discussed this already, that there is no definition of alert and action levels provided in the ISO bioburden document. And in a review of such documentations as PDA, both USP 1116 and USP 1231, and then also FDA guidance, there are some similarities. And so pulling from all of those documents, here is what we came up with as a good idea of what alert and level definitions are. Now, for an alert level, if you see your bioburden approaching that, you might have drifted away from normal operating conditions. Now, at this time, you may want to perform an investigation, but it's not required. And another thing to keep in mind that although you might see your bioburden repeatedly hitting that alert level, if you see that, you might look at that as if you were truly reaching an action level, because that might be some indication that something has changed and you need to take a look at it. Now, when it comes to an action level, now your process has drifted away from your normal conditions, and in this case, you must perform an investigation. Now, hopefully, that investigation will point you to something that you can fix, and you can implement corrective action and then move forward. Unfortunately, sometimes that doesn't happen, but an investigation does need to be performed. Now, one thing I'd like to point out here before I move on is that a lot of times you'll see both limits and levels referred to. And I just want to make clear that for our purposes, the term limit implies that the product has been impacted by an excursion above that value. Use of levels does not imply that the product has automatically been impacted, and that is the generally accepted term. So for everything that we're talking about here, except for one example when limits do come into play that Martel will address later, we are referring to levels throughout all of this. Now, here is a list of general concepts. We get a lot of questions from people about establishing levels, and these are some of the general recommendations that we have. First and foremost, don't base your product release or manufacture on bioburden values. Just because you've seen an increase doesn't mean you necessarily have compromised. You're to a corner and say, oh no, I've reached my action level, I've got to stop production. Don't do that. Another thing to keep in mind is that you'll see environmental data, and then you'll have bioburden data, and if you see an increase in one or the other, they don't necessarily go hand in hand. If you do see an increase in your environmental monitoring, don't automatically assume that you'll see an increase in the bioburden on your product. Environmental monitoring is meant to show that you have control over your environment, whereas the bioburden is coming from the manufacturer of the product, and if you're handling the product or it requires a lot of manipulation, that's most likely what is contributing to those counts and not something that has gone on in your environment. Another point to keep in mind is not setting your levels too soon. When you're a startup company and you've got new equipment, you've got new personnel, you're trying to get things set, your levels might not be the same then as when you actually get going into full manufacturing. And so you don't want to, once again, paint yourself into a corner by establishing levels that might not be truly representative of your entire process. Another key concept is don't establish the same levels for all of the products. As we've mentioned and will mention, if you've got a product that is using an overkill method, you have a little bit more flexibility in what your levels for bioburden might be as opposed to something that you're using a bioburden-based method. And also, all of your products aren't going to be the same. You might have one product that's nothing but molded plastic, and you may have another product that is made of metal components. So each of those things just inherently bring different things to the table, and so you need to keep that in mind when you're looking at your different products and your different manufacturing lines. And then last on this slide is looking at resistant organisms. This most generally comes into play when you're looking at a radiation sterilization modality, because your resistance does matter. And so not only do you want to know what kind of bioburden you have, you also need to take a look at the resistance of those microorganisms, because that might be important as well. Now, I've mentioned this a couple times, but here is a slide covering that. So what we are trying to get across on these couple of slides is that when you are using an overkill method, you're usually using a most resistant organisms, and you are inoculating a BI with 1 million CFU. Now, unless you have an incredibly dirty manufacturing area, your naturally occurring bioburden shouldn't be anywhere near 1 million CFUs. And when these cycles are developed, you use a half cycle, which shows that you can kill that million microorganisms, and then it's doubled. So you are looking at a 12 log reduction in your bioburden in those type of methods. Whereas if you get to a bioburden-based method, you can see a large degree of fluctuation in your bioburden, but here we're looking at what naturally occurs on your product. And so your alert and action levels usually have a bit more wiggle room for overkill methods and tighten up a little bit when you get to your bioburden-based methods. And I've mentioned this numerous times, radiation is a perfect example of that. And what is listed at the bottom of the slide here is that there are some different radiation validation methods that you can use. They are listed here. There's the VD-max method, there's the method 1, and the method 2. Now, when you're using the VD-max method, there are tables out there. They range right now from 15 kilogray to 35 kilogray as your sterilization dose, but each of the tables that correspond to those doses, they have a limit to them. The most common one is VD-max 25, and that table maxes out at 1,000 CFU. So right there you have a limit that you can introduce into your process of setting your levels, because it gives you a cutoff point. And for example, the 35 kilogray table goes up to 440,000 CFU. So you can see where there's variability in the limit that you're allowed, but there is an actual number that you can associate it with. Then you get to the method 1, and that goes up to 1 million CFU. So in some ways, that compares to an overkill method, but as your bioburden increases, then your dose increases. So in the VD-max, you have the ability to say, oh, I need to make sure that my bioburden doesn't get over this number. With method 1, that number keeps going up and up and up, and you don't necessarily want to go, oh, okay, the limit of that table is 1 million, so I'll use that. That doesn't necessarily make sense. You do need to, at that point in time, have some hard and fast numbers for you to look at that don't reach the limits of that table. And then when it comes to a method 2, it's a little bit different than the other methods in that instead of looking at your numbers of bioburden, you're actually looking at the resistance of the bioburden on your product. So in that case, you have even less flexibility because you're looking at your bioburden and your resistance, and if there is a change in that, you very well could be affecting your sterilization dose. Now, one thing that all of the requirements out there do say is that the manufacturer is responsible for trending and setting bioburden and environmental alert and action levels. And as I mentioned previously, the reason that you're doing this is that you need to use this as a means to monitor your manufacturing process and not a standalone product acceptance criteria. Now, before we get moving to our thoughts on how you should use your data, I want to point out some of the differences between what you're looking for in environmental monitoring versus bioburden testing. Environmental monitoring is looking at the control that you have over where your product is being manufactured, being it just a controlled environment or even a clean room. These same concepts mentioned on this slide all apply. You want to monitor the effectiveness of your cleaning and disinfection, and this usually is referring to your surfaces, your counters, your floors, your walls, your ceilings. And then you also want to look at the air quality in your area. If you're piping in dirty air, that's going to set the stage for you having some high counts in your air quality, but you want to be monitoring those. Given all the wildfires we've had around here, things like that can affect your air quality, and you would need to know that, and the only way you would know how it is affecting it is by doing this environmental monitoring. At the bottom there is listed water quality. Water becomes very, very important if you are using it in your manufacturing process. It is a breeding ground for microorganisms, and many, many times we will find that if there is a problem with your bioburden and you have water in your process, that's one of the first places we look, and a lot of times that's where you find the problem stems from. And the last thing listed on this slide is monitoring the effectiveness of gowning and the training of your personnel. If you have a product that does require manual manipulation, we will find that that's typically where your bioburden is coming from. So you need to make sure that your people are properly trained, and that they are using the best technique possible in putting your product together so that it does not lead to increases in your bioburden. This slide here, when we at Nelson refer to bioburden, it is, in our eyes, usually this definition: the population of viable microorganisms in or on a product in a sterile barrier system. As a medical testing facility, we are always testing these devices, and so we tend to just have the mentality that if we're talking about bioburden, it's always product bioburden. So I'll apologize if that causes any confusion in this. Overall, we are talking about environmental and product bioburden, but sometimes we might regress, and bioburden will be referred to as just the product bioburden. This definition obviously came from ISO 11737, and the important thing to really notice from this is that everything is reported as colony-forming units, or CFU. That's the terminology that is used. And as an example of what a colony-forming unit is, there's a membrane filtration slide there on the bottom left, where you can see the individual colonies such as those yellow ones there. And so we would go through and we would count each of those, and then you would get that reported on your report as how many CFUs that we saw on this membrane filter. What becomes a little interesting is that you see those blobs there, and we would still count that as one CFU, but what you can see on the bottom right-hand corner there is a microscopic representation of what that might really look like. And as you can see, it is potentially many individual CFUs, but the naked eye can't see that. So we do report it as one. Might be an underestimation based on what it truly is, but those are some of the limits of the testing that come into play, and nothing more so than when you see a bioburden spike. So right here is a representation of actual data, And when a bioburden test is performed, we usually are looking at aerobic bacteria and fungal organisms. So you can see that 10 samples were tested for each of these tests, both aerobic bacteria and fungal bacteria. We add those together, and then we take an average. And then once we have an average, we will adjust it using a recovery efficiency. A recovery efficiency is performed to validate your test of bioburden, and Martel will talk a little bit more about where recovery efficiency comes into play. But the things to note on this slide are that for both sets of data, the numbers are pretty typical, except for then you see that 697, and that definitely pops out at you as an increase in the bioburden. So spikes happen. Here's an example. And also what is seen here is what that can do to your data. The information on the right-hand side, your standard deviation turns out to be 40. But if you use a spike in your calculations, that bumps your standard deviation up to 209. So you can see where a spike can skew your data, obviously making it not normal, but you don't want to use bioburden spike data when you're starting to establish your levels, as it can skew you probably in the wrong direction. There is no standard definition for a spike out there. We look at spikes a couple of different ways. One common definition is that it's an individual value which is greater than or equal to twice the mean. Another way we look at it is if you see a log increase. Naturally, bioburden will just be variable due to its nature, so you will see a fluctuation of numbers, but it's those high ones that you want to keep out, want to keep in your sights. And if you do see spikes, investigate them. What you really need to know is if it's a common occurrence or sometimes it's just a one-time occurrence. Could it just potentially have been something went wrong on the manufacturing line, or potentially even something with that particular sample in the test method. If possible, you could do additional testing, but if you are seeing spikes, you definitely want to make sure that you can identify a problem and correct it, because you don't want those to keep showing up, and you want to be able to use data that doesn't have spikes to get your base levels established. This here is an example of a bioburden spreader. Now, spreaders typically occur if there's something on the product that might shed particulates. That gives the microorganisms something to grasp onto and spread across the plate. And if that's the case, this might just be one CFU, but because it had a means to spread, it looks like it might be a lot of colony-forming units. There are ways to minimize spreading in the test method, so make sure you are aware of that. Make sure the test method is made to take those into account and to try to minimize spreading. Also, spreaders can come from liquid samples where, if they are spread across the plate unevenly, once again, they just have this ability to spread across the plate. So once again, spreaders can't be used in establishing your data, and so you want to make sure that you have a method that does minimize spreading of your microorganisms. Another common example of things that we see in testing is shown here, and this is referred to as too numerous to count. And what you have essentially is a lawn of microorganisms, and there's no way to get individual counts from this. We refer to this as a TNTC. Once again, this is data that cannot be used when you are establishing your levels, because you obviously can't get a count off of it. If we see something like this, once again, there are ways to minimize this. You can test a smaller portion of the eluent, or you can increase the volume. You can perform, but once again, moving forward, the thing to keep in mind is that you need to have acceptable data to evaluate. No spikes, no spreaders, no TNTCs. And moving forward from there, I am now going to pass the time over to my esteemed colleague, Martel Winters, and he will give you some more information on how to actually use this data to set your levels. Thank you for your time.
Thank you, Wendy. In this slide, we talk about some things to consider when you are setting these levels. And so a few of these things are that when you are drawing samples for a bioburden test, they really should be pulled from various locations throughout the lot if possible, so that you have better assurance that those samples actually represent the lot itself from a bioburden standpoint. Most of the documents that we reviewed agree that you should gather data over some period of time, and a couple of them even mention that 1 year's time is appropriate. And that's likely to help us to account for potential variations that you'll see from different seasons throughout the year. It's also important as you trend your data, to review that, and a typical recommended frequency is yearly, so that you do have an established frequency where you're checking to make sure that you don't need to make any changes with your alert or action levels, or that you're not noticing a change that might not be wanted. Now, some of the documents and people we interact with sometimes would like to have temporary levels established fairly soon after manufacturing begins, so that they have something to look at in the first while, and that obviously is just fine. You'll want to make it very clear in your documentation that they are temporary levels and that they will be reestablished at some time in the future, so that when you make those changes, it doesn't look like you're doing it without good rationale. A couple other points on setting levels. We think it's wise that there be at least 4 sets of data that you're looking at. That gives you, in most cases, you're testing 10 samples in each set of data. So the number of sets is helpful because it gives you more data over time, and a higher number of samples, meaning up to 10 samples per set, it also helps you to get a better perspective of the consistency of the bioburden. We mentioned microbial characterization here. Those of you who have dealt with alert or action levels previously know that it is common when you hit certain levels, it is common to perform some level of microbial characterization, often identification of those microorganisms. And without a good baseline of what those microorganisms are, or what you tend to have on your product or in the environment, then to perform IDs when you have an excursion and then to look back, it doesn't help you very much. So as you are gathering this initial data for setting levels, it is also wise to do some level of characterization, be that a gram stain in colony morphology or an actual identification through genus and species. We also recommend that you employ a validated recovery efficiency on all this data when we're talking bioburden data. If each product has, or if each batch of data has its own recovery efficiency, that gives you a secondary variable which will make the data not appear as consistent. So if you have a validated recovery efficiency you apply to, whether you're applying the same value to each set of data, then that makes the data evaluation better. Wendy talked about standard deviations and use of standard deviations in establishing levels. We wanted to dig into that to some extent in this paper that we wrote, and we'll be talking about it here as well. Often people feel, in the industry, there are some who feel that that use of standard deviations in setting alert and action levels is not appropriate because of the general fact that bioburden tends to not follow a normal distribution. Now, the point that we want to make is that standard deviations are a great tool for understanding the dispersion of your data, the types of ranges that you tend to see. So whether or not the data fit into a normal distribution is less of an issue than the fact that standard deviations are a great tool for understanding that variability. Now, people also tend to default to saying that bioburden data fit a Poisson distribution rather than a normal distribution. And in the data sets that we evaluated, we actually found that Poisson was also not found. So in actuality, in most cases, neither one of them can be applied as far as a name to the type of data. But we still found the standard deviation approach to be very useful. So I have a couple slides here with a lot of data, and there are a lot of numbers to look at, and we don't expect that you have time to look at all. This is the same set of data that we use in the paper. And in the example here, you see we have month 1, month 2, and month 3. So the idea is that as we initiated manufacturing, we wanted to do bioburden sampling more frequently. And so in this example, we set up some bioburden testing once a month. You'll notice that we are employing the same recovery efficiency to each set of data. And you see we have calculated a bioburden estimate using that recovery efficiency. We have a standard deviation for each set. And then we also have a bioburden estimate plus 2 standard deviations and a bioburden estimate plus 3 standard deviations, as well as a bioburden estimate times 10. And we'll talk about those a little bit more, but we find those to be useful tools when you're evaluating data and establishing alert and action levels. Here's a second set of data where now that we have a baseline with months 1, 2, and 3, we now step back to a quarterly evaluation of bioburden. So here we have quarters 2, 3, and 4 with the same types of data evaluations below. Here's a summary of all that data where we have the overall average of 47.8. We have the recovery efficiency, and that makes an average bioburden estimate of all 6 data points or 6 data sets at 81.4. So the average standard deviation of those is 57.2. And to a chemist, that might seem high, but in the microbiology world, when you are dealing with medium to high levels of bioburden, like we were in these examples, then a standard deviation of 57.2 is actually pretty typical. So the average of the bioburden estimates plus 2 standard deviations is 195.8, and we also have the estimate plus 3 times standard deviation and the bioburden estimate times 10. We evaluated 47 different sets of data as part of this paper, and one thing we wanted to look at was the, as I mentioned before, the normality of the data. We found that of the 47 sets that we evaluated, that 39 were not normal, were not found to fit a normal distribution. And out of those 39 that were not normal, we found that 33 were not normal due to a single value being high that we're calling an outlier. And you might also call it a spike. They could both use those interchangeably here. And as we did this evaluation, Harry Bouchard, who Wendy mentioned was part of this team, determined a general rule of thumb for these values that tended to kick the data from a normal distribution to a not normal distribution. And that rule of thumb you came up with was if there was any one value in the data set which was more than the mean of the data plus 2 standard deviations. And that seemed to be a good rule of thumb that he determined as he looked through all these sets of data. Now, we also wondered if we made the sample size larger, if that would help move the data into a normal distribution. And in doing that, so all the data sets we evaluated were all 10 individual values. And when we grouped them into either 20 or 30 individual values, we found that the larger sample size did not make those normal. So the ones which were not normal remained not normal, even if we began to group the data together into larger sample sizes. When we talked about, she gave a good definition of alert level and action level, and so now we're going to provide some thoughts of things that you might consider doing when you hit these levels. Now, as Wendy mentioned, at the alert level, there's often no action required. Certainly no investigation is usually required. But we provide options here of things you can look at an alert level if you so desire. So the first is taking a good look at the trending of both the numbers and the types of the bioburden that you've obtained recently. And this can help you understand if this alert level we've triggered is a result of the bioburden increasing over time, or if this was a single event. Also looking at the identification of those microorganisms that from that data set that triggered the alert level. I can help you understand where those are coming from. And once you know the general types or the names of those microorganisms, you can then evaluate their general resistance to the sterilization process that you are using. You can also look at potential impacts from production personnel, meaning verification of proper training, or if new personnel have been involved. You can look for any process changes in manufacturing, or if there were also potentially changes in your sampling methods or testing methods. And the other ideas are looking at the laboratory, at their controls and monitors to ensure that the data that trigger the alert level is true data. Additional testing is always an option with current batches of the product to determine if it was a temporary event or if it's a signal of a longer-term situation. Cleaning of the production area is always a frequent action. Modification of the sampling plan, meaning perhaps beginning to sample either more samples or sampling more frequently. Checking for any changes in raw materials or in supplier. And as Wendy mentioned also about water often being a problem, taking a look at any water that might be part of your manufacturing environment. Now, these are a lot of things that we put under the alert level, and I want to make it clear it is very common at an alert level to sometimes only do one or two of these things, or sometimes even none of them, and just make note that indeed we triggered an alert level. Be aware, and we'll make sure that next time we test that the values are back down. But it is, in our opinion, wise to have a couple of things that you look at an alert level, and so we provide all of these, not assuming that you are going to implement all of these at an alert level, but there are things to consider. Now, when an action level is triggered, that's when it is common to consider doing most or all of the things that we've already mentioned under the alert level. And then added to that, at this point, usually an investigation is required to try and determine the root cause. And then also, it is often wise to then look at the potential impact that the alert level value that you obtained, the potential impact that might have on your sterilization specifications. When we're talking about that, usually we're referring to addressing the impact to the potential impact to the sterility assurance level. And usually when you're setting alert and action levels, you are setting them with the sterilization modality in mind, as Wendy mentioned. And hopefully these are normally established at points where you can trigger these levels and there still is no impact to your sterilization specifications. But depending on how much above the action level your value is actually reached, then you may notice that there might possibly be some potential impact. So it's wise to look at that at an action level. A few do's and don'ts for alert and action levels. We want to make it clear that it is not wise to establish a criteria where product is determined that product should not be released solely on triggering an alert or action level. Certainly alert and action levels are a part of what you look at when releasing product, but we try to make it clear it is our belief that you should not base product release solely on triggering, for example, an alert level. And also don't put into a procedure that you should discard product based on alert or action levels. This is especially the case if we're talking environmental counts. And environmental counts generally do not correlate to bioburden counts. So obtaining a high count on your environment certainly should not automatically, by itself, indicate that the product has been impacted. But something to consider, certainly, as I mentioned, is consider the impact on the sterilization process. That's the whole reason, really, that you are doing your bioburden testing or environmental monitoring, is to demonstrate continued control over microorganisms on the product, and so it is wise to have a procedure in place to help you evaluate what impact that count may have had. Okay, wrapping up now. We have an example here of bioburden methods, a bioburden-based method such as radiation. So in this case, we have a VD-max 25, so 25 kilograms sterilization dose, and the average bioburden in this example is 170 CFU. So in this situation, a good use of the information we've shared is that the alert level represents an average plus 2 standard deviations, and that the action level be set at an average plus 3 standard deviations. And at the average plus 3 standard deviations, there's still likely no impact to the appropriateness of your sterilization, cycle or the sterilization method, but it might be getting close, and so it is that seems to work well as a good action level criteria. Now, for VD-max 25, this is the one situation where we feel it's okay to use the word limit because, as Wendy mentioned, at VD-max 25, the table cuts off the bioburden of 1,000 CFU. So it makes sense, if you so desire, to have a bioburden limit where at what point you feel that there likely is some potential impact to your sterilization process. And this is actually discussed. The term bioburden limit comes from the radiation standard part 1, and the clause is referenced there. There's no strict definition for the bioburden limit in the standard at this point, but with VD-max, it seems appropriate to have that bioburden limit be the upper value of the table you are using. And in overkill methods, for example, ethylene oxide, steam, as Wendy mentioned, as part of that validation, you've demonstrated usually a 12-log reduction of the most resistant organism. And based on that, we feel it is wise to allow yourself some flexibility to establish your alert and action levels. Don't allow yourself so much flexibility that you have no control over the process. But so keeping that in mind, an alert level might be, for example, an average plus 3 standard deviations. And an action level might be the average bioburden times 10, which represents a 1-log change. And in this case, still, there is likely no impact to sterilization, but it demonstrates that you have concern over your bioburden count and want to demonstrate continued control over that bioburden. So in conclusion, just a few main points. Make sure that you're using actual bioburden to establish these levels and to review those periodically, and that the approach that you use should be tailored based on your product type and sterilization modality. And then also that the range of your bioburden is really more critical than whether the product fits into some type of statistical distribution. So we have our contact information there, and now we will take a moment to look through the question. I'll turn the time over here to Mike.
Okay, thanks, Wendy and Martel. We're very appreciative of your insights in this webinar. We've all gained some great information today. We've had lots of questions come in, and we're going to, Martel and Wendy are looking over them right now, and we're going to try to answer as many as we possibly can. If we don't get to your specific question, you do see Martel and Wendy's information there. They'd be happy to respond via email or telephone call as well if we don't get to your specific question. And with that, we will turn the time back over to Martel and Wendy, and we will go for about the next 10 minutes or so with questions that you have at this time.
Hi. I have a couple questions here I'd like to address. One of the first ones was addressed to me and telling me to speak up. I apologize for that. Apparently we were having a few technical difficulties, and it sounded like we were talking out of a box. I hope that they've been resolved. I hope that as the presentation went on that it was clear that both product bioburden and environmental bioburden are both addressed. It's just taking into account how you want to set those levels. One of the next questions we have is regarding someone specifically asked of applying a factor of 1.7 to mean bioburden counts to get the estimated bioburden. And is there a scientific rationale for this factor? Actually, in this presentation, we use the term recovery efficiency. Recovery efficiency is the same thing as a correction factor. It's just using the number in a different way, and you basically take 1 over your recovery efficiency in a decimal form, and then it will be transformed into a factor, a correction factor. And so that is how that is derived. And yes, it is very important to use that correction factor so that you are, like Martel indicated, eliminating another variable that might be there. Along those same lines, there was a question asking us how the bioburden recovery efficiency is determined. It's usually one of two ways. If you have naturally occurring bioburden, we try to use what is called the exhaustive rinse method, and what we do there is that after the product has been initially extracted, we remove all of the extraction eluent, and then we perform the extraction 3 additional times. So you have, and then it's just a calculation that is performed where you compare the number of organisms that come off on the first rinse, and what that tells us is that if you have bioburden that might be hiding in the nooks and crannies of your product, and you can't get everything off on that first rinse, that we take that into account to make sure that we are giving you a number that truly represents how much bioburden is on the product, and that's referred to as your bioburden estimate. There was another question about recovery efficiency, so I hope we have covered that. Let's see. Turn this over to Martel to address a couple as well.
There was a question we get frequently. came up today as well. It says, is there a limit at which you would link product release to bioburden in an EO overkill sterilization method? And the answer I usually say is a million CFU is the first thing to consider, because that's what you'd use a million CFU of the most resistant microorganism as part of your validation process. However, when you've reached that point, you're possibly no longer demonstrating much control over the bioburden, and control is an important part of being able to demonstrate some degree of control. So I usually recommend something less than 1 million, but certainly when you are in the range of anywhere between 1,000 and 10,000 CFU, even if they are the most resistant microorganism, which they usually are not, you still have plenty of safety built into that sterilization process. So I always recommend that there be some tie-in to the actual bioburden you've obtained, but also allowing lots of flexibility in an overkill process. Okay, there were a couple of questions on whether we should include outliers in your average for the calculation of your standard deviation. That's another question we get frequently. And the answer to that, in my opinion, is that it's up to you. If you include the outliers in your calculation, you will result in higher alert and action levels allowable, and also higher standard deviations or larger standard deviations. So if you feel that it is a normal part of your manufacturing process, based on the information you have, you can leave those in. However, if you take them out, the only danger is that you will result in lower averages and lower and smaller standard deviations. So certainly, if you feel you'd like to take them out, then, as far as I'm concerned, remove them, and then you're still safe, because those values will only be lower when you do that. Okay, let's see. The question on when it's applicable to perform testing for anaerobic bacteria. You notice perhaps in the data that we showed that we use aerobic bacteria and fungi in our evaluation. In almost every case that we have seen, whenever we perform anaerobic testing, the bacteria are actually facultative anaerobes, and that means that those same microorganisms are growing in your aerobic bacteria test. So if you're doing anaerobic testing, before you add those counts, do a check to make sure that they are strict anaerobes, and you just simply subculture and grow them aerobically to see if they will grow. We've got a similar question on spore count, and the same thing applies there. The spore test just tells you how many of your microorganisms can form spores or have formed spores. So the spore test gives you that percentage, but those same microorganisms are growing in your aerobic bacteria test. So again, to add those together, you're double counting the same microorganisms. And so a spore test helps you understand what percentage might form spores, but is usually not added to your overall bioburden count. There was a question about, for overkill sterilization methods, 100 CFU per device is the recommended action level. And this is going back in time a little ways, but back in the older versions of the ethylene oxide sterilization standard from AMI and ISO, there was reference that up to 100 CFU, there were no concerns and allowed you certain flexibilities. And that's just because of the vast amount of overkill that if your bioburden is below that level and you're using an overkill method, then certainly that would be allowable. Now, for an EO sterilization method, I can't imagine going lower than 100 CFU, but certainly going above that for an action level or even for an alert level would certainly be appropriate based on that level of overkill and the bioburden of the product. I'm turning the time over to Wendy.
I realized that I did not fully address the questions that we had about recovery efficiency. I did address an exhaustive recovery, and that is one method that can be used There were a few more questions that came in, talk about determining validated recovery efficiencies and whatnot. And generally in the industry, you want to have a recovery efficiency that is greater than 50%. If we do not get a recovery efficiency that's greater than 50%, there may be a couple of reasons for that. A lot of times, if you have a product with low bioburden and there are no organisms present, obviously we can't get any organisms off. In those cases, we would recommend that you perform an inoculated recovery efficiency, and that would be where we would inoculate your product with approximately 100 CFU of Bacillus atrophius. We would perform an extraction, and we would compare the number of organisms that come off with the number of organisms that were inoculated onto the product. So exhaustive recoveries, inoculated recoveries, either one is an acceptable means. You do want to have a validated recovery, and you do want it to be over 50%. If exhaustive recovery, typically we use manual shaking. If we do not get good recoveries using that method, there are different methods that we can use, such as mechanical shaking or maybe adding a sonication step to improve that efficiency. We have been told that we have time for one more question, so I will turn the time back over to Martel for that question, and I would like to thank you all for participating today.
I'm having a hard time choosing one of these questions here. There were a couple of questions related, again, to whether the bioburden distribution, whether it fits a normal or the question about whether it fits a log normal distribution. And in all the data we evaluated, we found that even though the bioburden data was not fitting into a typical normal distribution, we still felt that use of the standard distribution to help establish the bioburden range you're seeing was still appropriate. And so regardless of what statistical terminology you apply to your bioburden data, I would still recommend using the standard deviations as a great method to help establish your alert and action levels. And now I will turn the time over to Mike.
Well, unfortunately, we are out of time. And again, we'd like to thank you for participating in the webinar today, and we hope that you found the information valuable. The link that is available, that is showing on your screen right now, is a link to a white paper that is that Martel, Wendy, and team wrote that has been published. It was an article published in the June MD and DI issue, which is a shortened version of the white paper that is also available on QMed.com. And if you go to the QMed, you go to their resources link, and under the resources you click on the white papers, you will find this white paper. It was published back at the end of May 2013. So you can get this resource is available to you online. Again, we thank you for participating today, and we hope you have a great day.